everyone. Thanks for everybody who's joining today. So I'm going to talk about our work on olfaction, and primarily around the idea of bringing an atomic perspective on olfaction. But I want to argue that these may still serve as potentially druggable targets, and I'll get into that a little bit. So just as a broad overview, of course, my lab and why you're here is because you're interested in G protein coupled receptors, and what's fascinating and continues to fascinate people like me and Brian and Jeff and Tom is that these huge family of proteins, senses a huge diversity of stimuli, and that's responsible for allowing our cells and tissues to recognize what's happening on the outside and converting it to some sort of signal. And if we think about kind of like the role of GPCRs in sensory biology, it's pretty clear that GPCRs basically mediate three of our primary senses: our sense of smell, our sense of vision, and our sense of taste are all mediated by G protein coupled receptors. And among these sensory modalities, perhaps the best one that's understood to date is how our vision works. So just to recap a little bit, this is how, again, the fundamentals of vision, which is that at the back of your eye, there's a little set of cells, specific photoreceptors called rods and cones. And these little cells are highly specialized and tuned to respond to various wavelengths of light. For example, you have rod photoreceptors that respond to wavelengths of light kind of in this range, and cone opsins that respond in kind of these other wavelengths. And so the way the visual system is built is really a beautiful way of thinking about how the physics of light works and how our visual system has evolved to sense it. So again, here, for example, are the cells that enable this process. And zoom in a little bit, you can see kind of what these cells are made of. They're packed very tightly with this G protein coupled receptor called rhodopsin. And at a very deep level, we understand, for example, how a single photon of light can cause a conformational change in this chromophore retinal to drive activation of this protein rhodopsin. And on top of that, we know at a very deep level how the residues that surround retinal can tune the properties of an opsin to basically respond to, for example, shorter or longer wavelengths. And even more recently, really amazing work out of Gebhard Schertler's group has started to track the very, very early biophysics of what happens when a single photon of light strikes rhodopsin. This is just amazing work from these free electron lasers tracking, for example, what the conformational changes in retinal after 1 picosecond of light. So what I'd argue is that at a fundamental level, we understand our sense of vision really, really profoundly quite well. But by contrast, our sense of smell remains. Our understanding of our sense of smell remains somewhat rudimentary, and that's because, unlike the visual system, where our ability to perceive almost the entire set of wavelengths of light is mediated by only 3 receptors, our sense of smell requires 400 or so receptors. So this is, again, pioneering work by Richard Axel and Linda Buck now almost 30 years ago. This is just a dendrogram showing the hundreds of olfactory receptors that are in our genomes that enable us to detect and distinguish such a huge diversity of chemical stimuli. And just for what's going to be important later in this talk, these odorant receptors can be broadly categorized in two subfamilies. One are so-called fish-like odorant receptors, which are shown in this kind of orange graph here, or this orange set of sequences here, and then a much larger set of sequences that have exploded in tetrapods that once they started to walk around on land. And these respond to much more volatile odorants. So the fundamental logic of how this works for the olfactory system was proposed by Linda Buck and Richard Axel now almost, again, 25 or 30 years ago. And that's what's basically laid out in this nice little diagram here. So the idea is that we know that we can sense tens of thousands or maybe even hundreds of thousands of distinct odorants. And we only have 400 receptors. So clearly there can't be a one-to-one mapping that, there's a receptor for the smell of caraway and a receptor for the smell of menthol. It's almost certainly not as simple as that. And instead, what's more likely to be the case is that a given odorant can activate multiple receptors. And it's really the combination of receptors that are activated that gives rise to the perception of a smell. So for example, this odorant is specific for just this receptor, and it gives rise to a specific smell in the brain. And for example, this odorant, hits multiple receptors and it's perceived differently. So it's really the fundamental problem at hand is if we want to understand how olfaction works, we need to basically figure out this combinatorial logic to eventually one day be able to predictively say this is the chemical structure of a molecule and it has this kind of perception. Okay, so that's maybe the dream of what one would hope to be able to do. But the fundamental problem that's plagued the field is that unlike many GPCRs, which have started to become much more tractable for structural biology studies, biochemical studies, and certainly signaling studies, olfactory receptors for a long time have been really, really challenging to work with. And so, the fundamental goal that we're after is to decode this chemical logic of olfaction. We want to be able to map the structure of odorants onto the receptors and understand what that pairing is. But the fundamental challenges in the field have been that odorant receptors are incredibly poorly expressed. And this is thought to be because olfactory sensory neurons, where these receptors are express unique chaperones that either allow them to fold. And in fact, it's the folding deficits of these receptors that's really important for some of the biology around how the system gets set up. On top of that, most odorants, because they're volatile, can traverse through the air, are pretty insoluble. And on top of that, like most GPCRs, odorant receptors are very unstable, but on top, unlike many GPCRs, they're even more unstable because that's part of their biology. And we can get into that if people are interested. So we wanted to tackle this problem, really try to bring an atomic perspective to our understanding of olfaction. And the solution that we came up with in collaboration with Hiro Matsunami's lab at Duke was to work on so-called ectopic odorant receptors. So what I told you is that there's 400 or so odorant receptors expressed in sensory neurons in the nose, and these enable our sense of smell. But it turns out that there's this emerging literature that odorant receptors can also be expressed outside the nose, and these have really interesting moonlighting functions. So on the left here is just a single publication that just tried to quantify, for example, transcripts of a variety of odorant receptors in other parts of the body. For example, you can see this receptor OR51E2, highly expressed in the prostate. Others are expressed in different tissues. And people had seen this for some time, but over the past maybe 3 to 5 years, the actual biology of these odorant receptors has started to become more clear. So, for example, it's clear that OR51E2 expressed in certain prostate cancer cells might be important for driving certain types of functions there. Studies on another odorant receptor, OR6A2, in macrophages suggests that it's important in atherosclerosis. There's other work that suggests that enterochromaffin cells in the gut express odorant receptors that sense short-chain fatty acids, and this is responsible for sensing of the gut microbiota. So as a result, what we figured was that, okay, there's probably ectopic receptors that don't require these unique chaperones, and perhaps these might be a little bit more amenable for doing biochemistry and structural biology. The other trick that we thought about using was that, okay, so on the right here are all of the volatile odorants that we normally think of as odorant molecules, and these are very, very insoluble. Most of these are pretty insoluble. But it turns out that fish clade of odorant receptors recognizes highly soluble ligands, like these little short-chain fatty acids like propionate or lactate. So we figured, okay, maybe these would be a little bit more tractable than working on receptors that recognize these kinds of ligands. And then to get around the stability problem, we took an idea that was initially proposed by Henry Bourne almost 30 years ago, which is to split the heterotrimeric G protein into these so-called RAS domain and cleave out the alpha helical domain. This idea, has been really put into practice by Chris Tate's group at the MRC. And the simple idea is, again, that we take the alpha subunit, chop off the alpha helical domain. But here's the key thing: we basically fuse it to the C terminus of an odorant receptor. And the idea here is that this little domain, this little mini G protein, as Chris Tate's group calls them, basically stabilizes a GPCR in its active conformation. So we figured that this might be more amenable to structural biology studies. So with that, we have been tackling one receptor among these hundreds of receptors, a receptor called OR51E2. It's a receptor that, again, is ectopically expressed in a lot of different tissues and recognizes an odorant called propionate. And initially, we wanted to get a sense for whether this strategy of fusing this mini G protein to the C terminus had any utility. So here's just a simple signaling, a cyclic AMP signaling assay. You'll see that the potencies of odorants here are really, really abysmally low compared to what we normally think about for GPCRs. And so you can see propionate titrates with millimolar potency at this receptor. But if we make this mini G protein fusion, you can see that response is completely gone. And this may seem trivial to most people who work on GPCRs, but it really suggested to us that this mini G protein is coupled in a way to this receptor that is very productive. So with that, we were able to do some biochemistry, purify a complex of OR51E2 bound to all these various G protein stabilizing components that we've done, that we and others, many others in the field have started to do quite a bit. And eventually we were able to eke out a pretty tiny amount of this complex for structural biology. But with the amazing, advances in cryo-EM, we can take even tiny quantities of this material, and we're able to get a cryo-EM structure of this receptor, which is shown here. So let me just kind of walk you through it. Again, in green is a receptor. On the bottom are the various heterotrimeric G protein components that this receptor couples to. And then we can zoom in, for example, and see that this odorant propionate is bound within the site. Okay, so just kind of some high-level overview of comparing and contrasting the odorant binding site in this receptor to canonical GPCR like the beta receptor bound to adrenaline. One thing we noted is that this propionate is completely enclosed within this odorant receptor, and this likely reflects the fact that these odorants are very small, and in order to eke out any kind of potency, basically these receptors, at least this specific receptor, seems to completely enclose that the odorant. When we look at kind of the comparison to the beta receptor, you can see that in some ways the binding pockets, it's kind of in the same region as a classic GPCR orthosteric binding pocket, but it's really small, and it really kind of focuses on a little subset of the receptor compared to other receptors where ligands really start to take up a much larger amount of space. Okay, so with that, we wanted to start to get to understand some fundamentals of how odorants are recognized by their receptors. And so we, for this receptor, OR51E2, it's been known that this receptor is relatively narrowly tuned. The idea being that it's somewhat narrowly responsive to only short-chain fatty acids like acetate or propionate, but not responsive to, for example, longer-chain fatty acids. And here's some cyclic AMP data that shows that. And you can hopefully see on the right here that the binding pocket, which is outlined in gray here, is really quite small. It really doesn't tolerate longer-chain fatty acids. So, kind of a cute experiment that we could do is try to retune the response of OR51E2. And we figured as if we could, create space in the pocket by either mutating these two bulky amino acids to smaller groups, we might be able to retune the response of this receptor to other fatty acids. And so you can see, for example, creating a little bit of space at this position allows longer chain fatty acids like the octanoate, or for example, hexanoate to bind. And so what this starts to suggest to us is a couple of things. One, this odorant receptor, for example, has a very specific recognition motif. It has an arginine that recognizes this carboxylic acid that kind of pins part of this odorant into the pocket. But the rest of the selectivity determinant for this receptor is determined by the shape of the pocket, and changing the shape of the pocket can dramatically retune these receptors. Okay, so that's just how the odorants bind. How does that then lead to activation of the receptor? And I don't want to belabor the point here because this kind of can get in the weeds. But what I want to say is that we have, at least for this receptor and for the fish-like receptors, a pretty good model for how odorant binding turns receptors on. So we see in our cryo-EM structure again is this arginine residue that's coupled to this extracellular loop 3 of this receptor that engages the carboxylic acid of propionate. So we wondered, is this contact really critical? And obviously we could do the mutagenesis experiments. We see that if you mutate this arginine, that basically this receptor completely loses its response to propionate. We want to dig a little bit deeper. So working with Nagaraj and Vaidehi's group, we ended up doing a number of molecular dynamic simulations where we basically remove the propionate and then ask what happens. And what we see is pretty immediately this arginine, basically because it loses contact, this propionate, carboxylic acid starts to flop around in the breeze, and this leads to a pretty dynamic disorder in this region, this extracellular loop 3. And with Vaidehi's group, then we could design a gain-of-function experiment. We wondered, okay, if we substitute this glutamine for an aspartate in the pocket, could we basically pin back this arginine to lead to, for example, constitutive activity? So in simulations, what we predicted was that this arginine would engage this aspartate, and in signaling studies, we could show that, for example, just that mutation, that specific mutation, could lead to constitutive activity, suggesting again that it's very specifically pulling this arginine in that drives activation of this receptor. So it's still very, very early days. This is just one odorant receptor, and then it's again it belongs to this one fish-like family of odorant receptors. But we have a tentative model for how we think odorant receptors are activated by odorants. And so the idea here is that unlike many other GPCRs, which kind of have their own motifs, odorant receptors have this very unique FYGX motif that's in this central helix transmembrane helix 6. And our model is that this motif, as well as the top end of this of helix 6, along with this extracellular loop 3, are pretty dynamic in the absence of any odorant. And what happens when odorants bind is that they stabilize an inward movement of TM6 on the outside and an outward movement of TM6 on the inside, which allows the G protein to bind. Okay, so again, this is a speculative model. We don't have inactive structures of any odorant receptors. We don't have, we only have a single structure. So this obviously is a very working model. But if you'll allow me, I'll speculate a little bit on what this might mean for how odorant receptors evolve. And so because we obviously don't have access to structures of the broader odorant receptor family, we ended up looking at alpha-fold predictions for all 400 or so odorant receptors. And that's what's shown on the left here. So all those structures have been overlaid, and what's colored from red to blue is the AlphaFold confidence score. So red being AlphaFold feels very confident about the structure in that region, and blue being relatively poor confidence. And what I'll say is that the sequence level diversity across these regions on the extracellular side is actually pretty low. So these areas are not highly conserved across ORs, but their structure, the prediction is that their structures are highly conserved. The area where this breaks down is extracellular loop 3. And this region, AlphaFold, has a hard time predicting. And so our idea here is, and this is still a working idea, is that what happens in evolution is that this region diverges obviously in sequence, but also in structure. And that structural diversity is what allows odorant receptors to recognize such a huge diversity of small molecules. And this is analogous to basically how perhaps antibodies work, obviously by different mechanisms of diversity. But the loops of antibodies, specifically the third complementarity determining region, these are antibody structures, but holds true for antibodies too, is really the structural diversity of those loops that allows antibodies to recognize such a huge diversity of antigens. So we think there's perhaps some parallels into how structural diversity in proteins that are responsible for recognizing such a huge diversity of molecules arises. And for odorant receptors, it's again evolutionary diversity just by gene duplication and diversification in structure. For antibodies, it's obviously recombination and somatic hypermutation. Okay, so thus far I've told you about our work on a single odorant receptor, just perhaps that one right there. Really, if we want to get at this idea of understanding the combinatorial logic of olfaction, a single receptor is almost certainly not going to be enough. And really, what we'd like to be able to do is march along and understand hundreds of odorant receptors at once. But you can imagine that's going to be a really hard task. The vast majority of these receptors are completely intractable for structural biology. And so the approach that we've been taking with Hiro Matsunami's lab again is to use so-called ancestral protein reconstruction to try to get some insight into the other major classes of odorant receptors. So the idea behind ancestral protein reconstruction, and this is something that the protein engineers use quite a bit, is that if you have extant proteins that are relatively unstable, often if you can infer an ancestral sequence, these end up being often more thermostable, promiscuous, or evolvable. And often the protein engineers use this as a way to has good starting points for engineering new functions into proteins. So what Hiro's lab had come up with is a way of doing this for odorant receptors, and I'll kind of highlight how that works here. So this is all 23 receptors that are within the human OR151 family. So there's again, about a dozen or so families, and each one of them have about 20 to 30 members. So you can align all of these up, and that's basically what's shown here. We can zoom into, for example, the end of the region that comprises the end of TM3. These are the sequences that come up. And what Hiro's lab does is simply makes a construct that they call a consensus OR that keeps the most common residue at each position. So for example, you can see here's the highly conserved DRY motif in GPCRs, and it turns out that odorant receptors, at least this family, has an F at this position, so they basically keep the F instead of the canonical Y. So if we were to look at this as a dendrogram format, this is basically what happens. You have all of these individual odorant receptors, and these consensus receptors basically end up being the root of this tree of existing receptors. So again, these are somewhat Frankenstein receptors. These don't exist in nature, but what's important is that these receptors express far better than individual family members. So what you can see here is a cell surface expression experiment. In the dotted line here is just what happens if we transfect in the vector control. Each one of these gray lines here is all of these individual family members, or 51 family members. Hopefully I can convince you that these express basically not at all. Or 51E2, the one that we ended up getting a real structure, a cryo-EM structure of, is basically shown in green here. You can see it's one of the probably the best expressed OR in the genome in heterologous systems. The next best one, OR51E1, is expressed really quite poorly. This consensus receptor expresses really at very high levels. So this is a construct that's really amenable for structural biology, and with that we've been able to then get a cryo-EM structure of this consensus OR51 construct. And what's interesting about this receptor is that that we know it's constitutively active, and when we look at the cryo-EM map, there's basically no odorant bound in the binding site of this receptor. But with this consensus construct, we can then go back and ask, okay, how well does this consensus OR51 construct match the structure of our experimental structure of a truly human OR, which is OR51E2. So here again is just a structural comparison. Hopefully at this level you can see that as one might anticipate, these are almost identical at the secondary structural level. But we can now zoom into the binding pocket of this consensus receptor, again shown in blue here, and a truly native human receptor in green here. And I'll give you a minute to maybe compare back and forth, but hopefully what you can see is that by and large, the conformations of most of the side chains are conserved, and there are obviously some substitutions. For example, you can see this phenylalanine is an alanine in the native receptor, in this specific native receptor. But what we figured is that this at least gives us a good starting point to now be able to go model other members of the OR51 family. So we could, for example, take this consensus structure and make a nice homology model of a related receptor OR51E1. It's basically shown here, again, at a secondary structural level. And what's unique about OR51E1 compared to E2 is that this receptor responds to fatty acids that are a lot longer. So you can see, for example, here, this receptor can respond to pentanoic acid. Starting with now this homology model, we can go back and start to dock in a molecule like pentanoic acid and start to get some insight into how OR51E1 recognizes pentanoic acid and how that perhaps differs from OR51E2, which is now very selective for short chain fatty acids. Just to give you a flavor that we think this consensus OR strategy may enable us to start to get access to structure-enabled homology models where the closest structure that we're using has very high identity in the binding pocket. And so thus far, what I basically showed you is our work on kind of this family of ORs. We've in the past couple of months focused very heavily on getting consensus OR structures for many of these receptor olfactory receptor subfamilies. And just to give you a sense for what this looks like for now a completely new class of ORs, these are odorant receptors that respond to truly volatile odorants. And this is a much larger family, again, that's exploded in tetrapods. We've recently determined the structure of a consensus OR1, which is, again, one of these consensus receptors representing the OR1 family. And you can see here, this is bound to a more classic odorant like L-menthol. And what we're learning from this is that the molecular recognition for these volatile odorants, poorly soluble odorants, is really quite different from the story that I told you about propionate with this very specific carboxylic acid that gets recognized. So hopefully that gives you some sense for progress that we've made on understanding odorant receptors. We think that there's obviously a lot of work to be done to understand the chemical logic of the olfactory code. But in the short term, I think some of these ectopically expressed odorant receptors that have really interesting biology may end up being potentially useful new targets for other biology. So, for example, what I mentioned around OR51E2 in the gut sensing gut microbiota, or this receptor OR6A2 sensing molecules and driving atherosclerosis. So with that, I'll just give a shout out to the team, an amazing team of people that's been working on this for quite some time. This is a pretty deep collaboration with Hiro Matsunami's group at Duke, really a veritable expert in odorant receptor biology. And Vaidehi Nagarajan's group, who brings all of their amazing computational expertise. Christian Billisville in my lab has been really the main person who's been leading the charge on almost all of this work. Has been really a pretty challenging, as you can imagine, set of experiments. And he's been joined by a visiting student, Claudia Del Torrent. Claire DeMarche in Heroes Lab, and now has her own group, has been really one of the pioneers of this consensus OR idea. And she's been joined by Jeevan, really amazing undergraduate technician in Heroes Lab. And then in Vaidehi's lab, Wynand and Ning have been really driving our computational understanding of how this works. So with that, I'll stop and take questions. I'm happy to think about these both from the perspective of sensory biology, but also, as potential new drug targets.